Hello, my dear students. Good morning. Welcome to my channel and our new lesson. This lesson is all about seed selection. And at the end of this lesson, you will be able to apply some information, concepts, and principles relative to seed selection, of course, to your practical life if you are venturing in crop production and gardening. The topics which we will discuss in this video are reason for selecting good seeds before planting, characteristics of good seeds, methods of testing the viability of seeds, and computing the percentage germination. However, uh, before I will share to you information relative to this particular topics, I would like first uh, to have a brief review on the definition of said and give you short description of the different classes of seeds. All right, let's talk about a seed. A seed is defined as a mature ovule derived from pollination and fertilization process. Pollination is simply referred to the transfer of pollen grain from the anther of a male flower to the pistil of a female flower. And inside the pollen grain are male germ cells, and in the uh, female flower there is what we call ovary, where female germ cells are located. And when successful pollination happened, there will be a union of the male germ cell and female germ cell of the flowers, and fertilization comes in, and it will form a zygote, and of course once this will grow successfully, it will now form a seed. Let's talk about the classes of seeds. In the Philippines, insofar as my research is concerned, there are four classes, but in foreign countries they have five, with the inclusion of the uh, nucleus seed. Okay, but in our country, as what I've said, there are four classes, and these are the different classes of seeds. We have the breeder seed, the seed of a new variety that has the highest purity and is produced, developed, controlled, and provided directly by breeders or their institutions for further multiplication. And the progeny of this is foundation seed. That's why the description of the foundation seed is the progeny of the breeder seed produced by recognized seed producing agencies in public and private sector under the supervision of said certification agencies. And of course, we have also the registered seed. This is the progeny of foundation seed that is so handled as to maintain its genetic identity and purity according to the standard specified for the particular crop being certified. And this breeder seed, foundation seed, and registered seeds are actually under the supervision of the Bureau of Plant Industry and or Department of Agriculture, because there are several agencies attached to the Department of Agriculture or yeah Bureau of Plant Breeding. And we have also the last one is certified seeds, and the this is the progeny of foundation, registered or certified seeds handled to maintain sufficient varietal identity and purity, grown by selected farmers under prescribed conditions of culture and isolation, and subject to field and seed inspection prior to the approval by the certifying agency. So once the seeds produced by um, farmers will be certified, and this is now ready for distribution for commercial uh, distribution. And these are now the seed used for or by farmers, commonly used by farmers. Okay, let's talk about the reasons for selecting good seeds before planting. Actually, I identified only one here, which is very much important. It saved time, money, and efforts in replacement of plants. Naturally, if you will use poor quality seeds, there is a tendency that the seeds will, or the plants coming from poor quality seeds will not survive in times of adversities, like, for example, typhoon and many others. Okay, so that's the reason mainly why we select good seeds before planting. And as what I've said a while ago, there are so many other reasons. Perhaps there will be delayed in terms of harvest. There will be no uniform growth of your plants, and in the production alone of seedlings, if you are producing seedlings before it will be transplanted to the growing area, um, sometimes they are susceptible for plant diseases and pests. Okay, so that's how important really is selection of good seeds. Time, money, and efforts is or are wasted if you will not select good seeds. Let's talk about the characteristics of good seeds. We have number one, it must be genetically pure. That is in terms of variety. Meaning to say there is no mixture of other varieties of that particular seeds. When we are talking about variety, this represents a more precise defined group of plants selected from within a species with a common set of characteristics. Okay, example of this is, for example, corn. Then the varieties of corn, several, sabihin natin, subnames um, or subspecies. We have golden bantam, ambrosia, and hybrid. Uh, this these are some examples of the varieties of corn. Okay. Second, it must have a uniform size, weight, and color. Of course, this can be done through ocular or through critical sightseeing. Look closely the seeds which you will buy. If it's an open, uh, it's in an open container. But if you will be able to buy seeds in the sachet, of course you cannot do that because outright you will really buy it because it's you will not be allowed by the seed dealer or agricultural supply. Then we have it must be physically pure. When we are talking about physically pure, it must be free from stones, debris, dust, leaves, twigs, stems, flowers, fruit without other crop seeds and inert material. So there is no mixture of dirts in short. Okay. Then we have, by the way, this is very much common in large scale production. Okay. The last is it must be viable. Viability pertains to the capacity of the seeds to germinate under normal conditions. So before planting the seeds, you need to really determine how viable it is. So in what way? Have another. We have this methods of testing the viability of seeds. The plate method. This is the utilization of plate. So what is commonly done by farmers or even myself, I do this. I get a plate, then I get a tissue paper, and I will place the seeds at a desired number. Then I will pour water just good enough to wet the tissue paper. Then after that, I will cover the plate and place that in a dark, cool, and dry place. And after 3 to 4 days, it will be opened, and the number of seeds grown will be counted, and of course, the percentage germination will be uh, will be computed. So this is figure one, um, showing the plate method. There are other farmers using petri dish, but I guess that's so very, very hard to find if you have no petri dish, or very hard to test if you have no petri dish. So use the available materials at home. Then we have ragdoll This is ragdoll method. This is not ragdoll plate. This is ragdoll method utilizing the rag in order to test the to determine the percentage of germination. You will lay the activity here is you will lay down the moist rag uh, in the floor. Then you will place several seeds like this, and then there is a stick, and you will roll around this stick, and this is now the the picture or the image. So up, you will also place this in a cold dry place, and after 3 days you will open this, and of course you can see that there are seeds germinating. And upon opening, you will count the number of seeds grown, and the percentage of germination will be computed. And of course, with the last one, seed box method, this is somehow laborious. This time, it's not anymore um, applicable. During our days, when we were still in college, we need to prepare seed box with a height of 7.5 or 7 centimeters, 33 centimeters width, and length is 50 centimeters, and there will be 48 seedlings which can be planted in this particular size of seed box. But today, we are not anymore using this method. What's important is we will be able to really determine that the seeds has the capability to grow. And of course, just the same, you have to place the seed box in a safe place, because there are snails, there are ants who will or which will possibly destroy your seedlings, or they might eat your seeds in the seed box. Okay, so once the seeds will grow after 3 to 5 minutes, at 3 to 5 days. Of course, the number of seeds grown will be counted, and the percentage germination will be computed. And we have here the computing the germination percentage. This is the formula. Um, Percentage germination (PG) is equals to the number of seeds sown, then number of seeds grown times 100. And of course, given this example, if you will sow 100 seeds in the rug or in the plate or in the seed box, and there will be 78 seeds germinated. This will be divided by 100. then times 100. I think the result is 78%. Of course, 78%. In order for you to be guided on whether or not you will continue to be using the seeds where you get this 100, or where you the seeds used in testing the viability of seeds. For me, to make sure that you will not encounter problem in the replacement, 90% is very much okay. Sometimes we give allowance until 80%, but below 80%, it's not anymore good to use these seeds tested. Why? Because you are already sure that there will be, for instance, in this example, there will be uh, 22% mortality, assured mortality. Okay? If you will use the seeds in the field for planting. So it's necessary at least 90% or 80% can be allowed. Others use 80% in terms of scarcity of seeds. Okay? So thank you very much, and God bless you all. Good morning once again.